Good morning, everybody. Thank you for having me here as keynote speaker at the International Conference on Research in Life Science and Healthcare, held on 12th of May 2023 at Berlin, Germany. It's sponsored by Eurasia. My presentation today is entitled Rusip as Source of Prospective Marine Bacterial Enzymes for Food and Health Industry. This presentation is based on my latest research conducted in the last 4 years. Let's move to the next slide. Let us start with the background. I would like to emphasize the importance of enzyme, particularly proteases in food and health industries. I've shown here in this slide the hypothetic structure of protease as a macromolecule. The roles of proteases are so many in the industry, but at least there are 4 that catch my interest. First, protease is largely used as meat tenderizer. Second, protease has been widely and commercially used as anti-thrombosis agent or drugs. In the market, for example, we can find nattokinase, staphylokinase, and streptokinase. These 3 anti-thrombosis agents are better in comparison with heparin because enzymes provides very low side effects. And then third, protease is the main hydrolytic enzyme produced by indigenous bacteria in biomedical waste with potential as bioremediation agent. I conducted this research in 2018. And fourth, protease has potential to be used as anticoagulation diagnostic reagent, for example, in the Lee-White test, involving blood sample. Protease can be a substitute for EDTA or ethylene diamine tetraacetate in case of blood sample of patient with allergy sensitiveness. Because protease is important, there is need or high demand to obtain new source of protease. This is actually to make protease more available and therefore will be more economic, the price getting lower if they are more available. Bacterial protease especially is preferred to be used in industry because it's more renewable compared to protease from plant or animal. Why? Because bacteria are fast to grow, they are easy to manipulate and control in the context of enzyme production in industry. Proteases that have high values are those having stability in harsh conditions, such as too high or too low temperature, diverse pH or acidity, and too low or high pressure. Such conditions are common in any manufacture process. Enzymes generated from the sea, in particular, including marine bacteria, offer high stability because they are adapted to harsh or extreme environment of the sea, such as high salinity, varied temperature, and pressure. Proteases that have high value in food and drug industry are the food-grade ones, preferably produced by non-pathogenic bacteria to ease the handling process. Overall, valuable proteases are those having high activity too. In unit, 1 unit equals with 1 micromole per minute, supporting its function as catalysator in biochemical reactions. Let's now talk about Rusip. What is Rusip? Rusip is fermented fish sauce, commonly produced and consumed in South Sumatra here in the map, and Bangka Belitung. It's usually made of marine fish with high protein. However, we have discovered that Rusip can also be made from any edible marine animals, not just fish. The fermentation process in Rusip involves protein degradation by proteolytic bacteria. Therefore, it makes sense if we isolate proteolytic bacteria from Rusip. Yeah? Usually, the fermentation process takes 24 hours at least. It can be extended to 7 days. Rusip is the subject that I want to introduce in this speech today because of several advantages it offers in the effort of discovering new marine bacterial protease. Protease from proteolytic bacteria in marine fish Rusip has potentials as follows. First, it has high activity. Most important characteristic of enzyme is high activity because of its role as biocatalyst. And then it is preferred that the protease is food grade, and Rusip can produce that because Rusip is food, of course. Yeah. And then third, stable in high temperature, and this comes from the fact that the enzyme is generated by bacteria of the sea. Yeah, they live in undersea environment with high temperature sometimes or low temperature sometimes, and also stable in high acidity or pH. Yeah, because in the sea we know the water has high salinity, high content of salt. Yeah. And then stable in high pressure. The sea environment are exposed to high pressure. Okay. The objectives of my overall research involving Rusip is first to isolate new enzyme producing bacteria from marine fish or marine sea cucumber Rusip, and then to apply the obtained enzyme in food and health industry, leading to commercialization of bacterial enzyme from Rusip. Yeah. So we expect to obtain new bacteria, new drugs, new enzymes, new food ingredients. The general method to obtain commercial bacterial protease from Rusip, based on our studies, is described here. The steps include first, Rusip preparation, the making of Rusip from diverse marine fish or marine animals. This requires a fermentation process from 1 to 7 days. This step is followed by isolation of enzyme producing bacteria. Usually we use skim milk agar to isolate proteolytic bacteria. We can use oil-based media to isolate the lipase producing bacteria, or we can use the cellulose based agar to obtain or to isolate the amylum producing bacteria. After we obtain the bacteria, and then we will conduct the enzyme extraction. This involves centrifugation, and also sometimes we have to conduct the prolonged incubation to obtain more enzymes. And then this step is followed by enzyme purification and characterization. This step usually involves SDS page, and then chromatography, and etc. Yeah, all related with the purification of protein can be applied to enzyme. And then the last but not least is the enzyme application. We need to apply the enzymes that we obtain to create innovative products. Several of them are methanizer, and then bioremediation agent, and then anti-thrombosis agent, and anticoagulation agent. As far as our concern, several promising results could be obtained by exploiting Rusip of sea, fish, and animal. In this case, the sea cucumber. What I have shown here as example of the sea animal fermented into Rusip, from sand sea cucumber or Holothurias scabra. This kind of sea cucumber is interesting because it has the highest level of proteins compared to other types of sea cucumbers that we collected. To date, my research team is able to produce and scale up bacterial protease from Rusip of this sea cucumber and develop it as a wound healing agent. And currently, the clinical trial phase 3 is ongoing prior to commercialization. Our product has brand Debrizim. And in this picture, the improvement of a wound when treated with our protease can be seen here. The role of our protease is as the debridement agent to clean up the wound, and that will make the healing become faster. The next result in this slide is the use of bacterial protease from sea cucumber Rusip, which has also been successful to be used as methanizer. The bacterial protease was used to submerge meats of different types. As we can see here, there are 3 types of meats. First from beef, the second one from chicken, and the third from tuna. The bacterial protease that was extracted from bacteria isolated from Rusip of sea cucumber was used to submerge these meats, and then the meat tenderizing activity was monitored or observed by the denaturation levels of eosin and myosin. As we know, both are fibrous proteins which cause meat to become hard. Bacterial protease in this case can degrade these fibrous proteins, and as the denaturation and degradation of both proteins occurred, the number of protein bands of smaller size will appear on SDS page gel. So we use SDS page method to measure the activity of tenderizing protein by protease. Here is in the right slide, on the right side of my slide. Here are the conclusions. Based on our studies, proteases from marine fish or sea animal Rusip has high activity and other leading characteristics suitable for food and health applications. As proof, we can develop protease from bacteria isolated from Rusip of sea cucumber, have the potential to be used as methanizer and also as a debrizim agent of wound, that will make the healing process faster. Here are the references, all of the publications that I have used to describe my presentation today. So these are published by mostly my students, and I have been the corresponding authors. Okay, finally, thank you for all of your attention and appreciation. This speech is presented for Berlin International Conference on Research in Life Science and Healthcare, May 12, 2023. I hope you all are happy and healthy.